Hello, everyone. It's really my honor to stand here to present my last work. Today, my name is Jie Liu. I'm a postdoc at the ABI Lab from Sargent Uni Adelaide. So today I'm going to talk about my recently proposed computation method called DegradeMaster, which can accurately predict the degradation ability of a small molecule called PROTAC. Yeah. So as many of us are aware of that the protein degradation is a very highly regulated cellular process, and it's mediated by the ubiquitin polysome system. So the target proteins will first be tagged with the ubiquitin chain via the E1 enzymes for activation, the E2 for conjugation, and finally the E3 ligase for the destruction of the target proteins. So what is PROTAC? PROTAC is a small molecule that can hijack this process, that can induce the degradation of the disease-causing proteins, which would otherwise be stable. So this makes PROTAC plays a very critical role in the cancer drug discovery. So the basic structure of the PROTAC usually consists of three components. So there's a warhead, also known as the target binding ligand, and there's a E3 ligand that will recruit the E3 ligase, and there's a linker that connects these two ligands and forms a stable molecule. So during the hijacking process, the PROTAC will first bind with the E3 ligase and the target protein simultaneously, and forms a target protein PROTAC E3 ligase ternary complex. And then the PROTAC will cause the target protein to be polyubiquitinated, and then followed by the destruction of the target protein. And the best part about this process is that the PROTAC molecule can be recycled and continue this process again until the target protein is fully destructed. So given the critical role of the PROTAC molecule, there have already been a lot of trials for the biochemists that trying to design and generate some novel PROTACs. So the conventional PROTAC design pipeline is a trial and error process. As you can see here, the biochemists will first choose some ligands from the PROTAC database. So there is already a PROTAC database that have the existing warheads and the E3 ligands. They first choose these ligands. Just a minute. Here we go. So they first choose these two ligands, and then they try to design the linker that can connect these two ligands. So usually they can now just have the best shot at the first time. So after designing a linker, they'll conduct experiments and try to observe if this PROTAC is good enough as a degrader. So they usually observe the IC50 and DC50. If the matrix does not go well, they'll go back to the linker design process and trying to optimize the length or the chemical bonds of the linkers. And hopefully after repeating this loop multiple times, hopefully they can find an optimized linker and get a designed PROTAC. So as we can see from this pipeline, the conventional PROTAC design is a very cost-intensive process. So it usually requires a lot of expert knowledge of the chemistry and molecular biology, and to get a proper linkers, you also need a lot of laboratory trial and error process. So a very natural idea is can we design a machine learning or deep learning model, or use a computational method to automatically to predict the degradation ability of a PROTAC. So thanks for the accumulation of the over 20 years, the high quality data of the PROTAC, and the recently proposed PROTAC DB has accumulated over 8,000 PROTACs and over 500 warheads and over 5,000 linkers. So this accumulated data makes it possible for us to build a machine learning model to try to predict the PROTAC. And before our work, there actually have been some deep learning models proposed, but these models suffer from various limitations, which we'll talk about later. So we use this data to build our own computational method called DegradeMaster that can accelerate the original design of the PROTAC molecule. So this is the overall framework of our model. I'm going to go into the details of each module. So first, we can see the input and output of the model. So the input is the ternary complex, which consists of the target protein, the PROTAC, and the EV ligase. And our model will will give a binary score for each ternary complex. So the 0 will indicate very low chance of this PROTAC that induce the degradation of the target proteins with this E3 ligase. And the 1 indicates the very high chance of degradation. So as we can see, we first use a 3D graph structure to model the target proteins, the ligase, and the PROTAC. So for example, for the target protein, each node will represent a residue, and each edge will be measured by the distance between these residues in the 3D space. And for the PROTAC, we use the node to represent the atom, and the edge will be the chemical bonds between these atoms. So after constructing the 3D graphs, we input these 3D graphs into the corresponding encoders. Here, we designed E3 equivalent encoders corresponding for each 3D graphs, which can capture the 3D structure information of these graphs. And and also these 3 encoders have independent parameters as well. So after encoding these 3D graphs and convert them from the sparse high-dimensional structure into a low-dimensional dense embeddings, we designed a pooling module here to combine these 3 embeddings together and through an MLP and finally get a score. All right. Yeah. So before keep on going to the details of the model, I'll introduce how we construct our data set. So as I talk about, there's a database called PROTAC-DB that accumulates all the PROTAC molecules. So we extract the PROTACs from the PROTAC-DB and get smiles. As for the target proteins and the EV ligase, we turn to the PDB database and to extract those that have crystal structures. But for those that do not have crystal structures, we use the AlphaFold-DB to get the predicted structures and add them to our training dataset. All right. And besides the structure information of the proteins and molecules, we're also trying to add some attribute information into our model. So as we can see, there are some raw features. These are basically the the physicochemical properties of the PROTAC molecules, such as like molecule weights, the X log P3, the ring counts. So we collect over like 20 raw features, and but these raw features, not all these raw features are very useful to the final prediction of the degradation. So we design the feature selection module here. So we first use a filter method, which trying to calculate the statistical distribution of each raw features and set a threshold. So if the features are below or above these thresholds, we just filter them. And then we design an embedding method, which uses GPT importance to measure the importance of these raw features as well. So after these 2 field selection, we finally get 6 selected features, which we think is very important to the prediction of the degradation. And we add these selected features into our model. Yeah, this is the selection results here. So as you can see, for each ternary complex, we will have a adjacency matrix, which indicates the structure information of the atoms and residues. And we also have a coordinate matrix, which represents the 3D coordinates of each node. And we also have a feature matrix, which is the selected features. All right, so we input these 3 metrics into our model. And one major innovation of our model compared to the previous baselines is that we find that even though the PROTAC-DB now have over 8,000 entries, in total, but only like 20% of these entries are labeled. So by label, I mean we know the ground truth labels of the if this PROTAC is a good PROTAC or bad, is a good degrader or bad degrader. But the rest 80% of the PROTACs are unlabeled, so we do not know they are good or not. So the previous models will just get rid of this unlabeled data, I think, because they are fully supervised model, and they just get rid of those data that do not have labels. But what we think is that probably because these PROTACs, even though they do not have labels, but they do have the structure information that can help the models help in the training process of the model. So here we design a pseudo labeling strategy here. So the basic idea is first we use the label PROTAC entries to pre-train our DegradeMaster model. And then we use the pre-train model to predict the labels of the unlabeled data. So we give them labels. So these labels are pseudo labels. But we are not gonna use all these pseudo labels. We're gonna choose and select those that are most promising, that can we think the most promising to help the enhance the model performance. So here we design a pseudo label selection strategy here. After the selection, we give the most promising pseudo labels, and we add these pseudo labels to the original label sets and combine together, and retrain our DegradeMaster again. So with this process, we acquire the best performance compared to all four baselines. Here we choose two machine learning models, which is SVM and random forest. And we also include two recently proposed deep learning model, the PROTAC and PROTAC-STAN. So our model's performance beats all these baselines on accuracy, accuracy, and efficiency score, and the performance gap is really, really large. So yeah, as you can see, this is the RSA curves as well. Okay. Another interesting part about the model is that we design attention strategy in our encoder. So here we'll try to visualize these attention weights. So we project the attention weights back into the 3D graphs, and use the density of the color of each chemical bond to to indicate the attention weights. So what we observe is that the atoms at the the Warhead ligand interface will receive higher attention than those that is far away from the interface, as you can observe here and here. And as for the linker, the chemical bonds and atoms at both ends of the linker will receive higher attention than the chemical bonds in between. So this means that our model can tell, for each PROTAC, our model can tell which part of this PROTAC plays a more important role in the degradation prediction. All right. And we also test the model efficiency here. So we compare with the three deep learning models, and our model achieves the second to the best training time efficiency, even though our model has much more parameters than the PROTAC baseline. And here I use a very simple example to show you how to use this DegradeMaster. So all you need to prepare is first this Excel file, which has the file names for the target proteins, the ligase, and the smiles of the PROTAC. And then you prepare the PDB files or the ML2 files for the proteins, and and prepare the files, and then just run this command. It's quite simple, and you get these results. So for each PROTAC, you have the prediction scores and the prediction probability of this PROTAC corresponding to each target protein. All right. Here I use a case study to illustrate how our model can accelerate the design of the PROTAC molecule. So BRD7 and BRD9 are two proteins that play a very crucial roles in cancer. So these two proteins, for example, the BRD9 are very commonly found overexpressed in cervical cancer and non-small cell lung cancer. And for the BRD7, it is downregulated in breast cancer. So over the years, the researchers have been developing PROTACs that can efficiently degrade the BRD7 and BRD9. So unfortunately, there's a PROTAC called VZ185 that can very efficiently degrade this two target proteins. So the researcher designed choose a BI7273 as a warhead, and they choose the virtual ligand as the virtual ligand, as we talked about before. The researcher designed 16 different kinds of linkers that connects these two ligands, right? So as we can observe here, the linkers have different lenses and different control compounds, and so the researcher conduct experiments on these 16 linkers and have the results here, which is the rightmost column, as we can observe here. This say the 15% threshold as the 15% remaining protein as a threshold. So if the remaining protein is below this threshold, this PROTAC candidates will be considered as a very efficient degrader. Otherwise, it will be considered a bad degrader. So we use red color to indicate the bad degrader and blue color to indicate a good degrader. And in these three columns, these are the results of the prediction results of the computational methods. So this column is the prediction results of our model. As you can see here, the prediction results of our model has the best match with the ground truth experimental results, with only 2 false positive and only 1 false negative. So this can demonstrate that our DegradeMaster model can truly help to pre-select the more promising PROTAC candidates in the PROTAC design process. For example, sorry. For example, we can see that our model has the better prediction results for the C6 and C10. And this can accelerate the PROTAC design process and save a lot of time and costs. And for those that have interest in our model, we provide our code in the GitHub, and we also uploaded all our training dataset to the Zendode. So you can download our dataset and our code from these websites, and very easily to repeat our model. Yeah. And I want to thank my people from my teams and collaborators that helped me a lot in developing this model. I want to thank Phil Yi, who's my supervisor, and introduced me the PROTAC, and hopefully we can do better in this project in the future. And I also want to thank Luke and Michael. They helped me a lot in the paper revision and the polishing process. And yeah, that's pretty much what I'm talking about today. Thank you.
Thanks. Okay. We'll have time for one question. Number 4.
I have a question. I understood from the presentation that the model
Yes.
designs a different linker.
You can stay by the mic. That's better.
Is that there is kind of reverberation. Essentially, I was wondering if it is possible to apply the same technology for the design of the ligand.
Sorry, I didn't quite catch it. You mean the design of our model or what?
The model improves the design of the linker, right?
Yeah. The accelerate the design of linker. Yeah. We do not generate the linker, but we predict if the given linker is a good linker or bad linker for the PROTAC. Yeah.
Is it possible to improve the linker so the specificity of the degrader is better?
Because this is only a prediction model, what we are trying to currently do is trying to develop a generation model that can generate a given a linker with given lengths and the given chemical properties. So that's what we're trying to do in the next step. But Unfortunately, for this model, we cannot improve the structure of the linker, but we can only predict if it's good or not. Yeah.
Okay, thank you.
Yeah. Thank you.
So I'd like to thank again all the speakers of this session and you for your questions. And we'll reconvene at 2.
Thank you.